Welcome, enthusiasts. Today we're embarking on a journey that marks the very beginning of a plant's life, seed germination. In this video, we'll explore the intricate and magical transformation process of a tiny dormant seed into a thriving cannabis plant. Sit back and relax as we discuss the entire cannabis seed germination process from imbibition to planting. Whether you're a novice grower or an experienced cultivator, our educational guide is designed to help enthusiasts achieve a successful germination and set the stage for a bountiful harvest. Before we begin, don't forget to like, subscribe, and hit the bell icon to stay updated with our latest videos. Also, please remember that this is an educational video, and therefore all information provided is for informational purposes only. Now, let's begin. Seed germination is a fascinating and intricate process that marks the beginning of a new plant's life. It all starts with imbibition, where the seed absorbs water, causing it to swell and soften. This initial hydration is critical, as it activates enzymes that kickstart the seed's metabolic processes. As the water permeates the seed, it also allows for the uptake of oxygen, which is vital for cellular respiration and will help the seed spring to life. Once the imbibition process is complete, the seed's metabolism goes into overdrive. Enzymes begin converting the seed's stored food reserves, such as starches, proteins, and fats, into energy and essential building blocks for new cells. This metabolic awakening fuels the rapid cell division necessary for growth. The first visible sign of this transformation is the emergence of the radicle, or embryonic root. The radicle's sole purpose is to anchor the seed in the soil and start absorbing water and nutrients. As the radicle digs into the soil, the shoot begins to emerge. The hypocotyl, a stem-like structure below the seed leaves, also known as cotyledons. The stem then elongates and pushes the seedling above the soil surface. As the cotyledons unfurl, it provides the seedling's first photosynthetic activity. This early photosynthesis is crucial, as it generates energy to sustain further growth. With the seedling now above ground, true leaves start to develop, more adept at capturing sunlight and continuing the photosynthetic process. Several factors influence the success of seed germination. For example, adequate moisture is essential for imbibition and enzyme activation, while oxygen is necessary for respiration. Similarly, the temperature must be within an optimal range specific to each plant species. Too hot or too cold conditions can hinder germination. Light also plays a role, as some seeds require light to germinate, while others need darkness. Additionally, some seeds have dormancy mechanisms that must be overcome through processes like scarification or stratification. Understanding these germination fundamentals is crucial, especially when discussing the cannabis plant, where the quality of the seeds directly impacts the final product. In the quest for high-quality cannabis, selecting the perfect seeds is akin to laying the foundation for a masterpiece. Here are some considerations growers think about then choosing cannabis seeds. First things first, cultivators always choose their seeds wisely. They look for reputable seed banks or breeders known for their reliability and commitment to genetic stability. Reputable seed breeders offer an advantage to more novice growers, as they should also provide valuable information such as strain details, growth patterns, flowering times, and anticipated yields. Armed with this detailed knowledge, cultivators can explore the different types of seeds available that best suit their cultivation goals. Now let's talk about assessing seed quality. Appearance speaks volumes. Cultivators seek seeds that have a rich dark hue, typically shades of brown or black, with a lustrous sheen. But they'll steer clear of seeds that are green, white, or pale, as these are likely immature and may struggle to germinate. Next up, texture. Cultivators gently squeeze the seeds between their fingers. High-quality seeds should feel firm and resistant to pressure, signaling robustness and vitality. If a seed yields too easily or crumbles, it's likely past its prime or compromised in some way. Size and shape are also important indicators. Healthy seeds are usually plump and generously sized. Experienced cultivators are cautious of seeds that are small or misshapen, as these might signal inferior quality and reduced potential for successful germination. Cultivators will also consider how the seeds were stored. Ensuring the chosen seeds have been meticulously stored in cool, dark, and dry conditions to preserve their viability is crucial. The seeds subjected to unfavorable storage conditions are prone to diminished germination rates and may fail to thrive. Additionally, cultivators will consider the genetics of their chosen seeds carefully. Selecting varieties that align with their goals, whether prioritizing medicinal aspects, specific terpene profiles, or particular growth characteristics, can set the stage for a successful and fulfilling growing experience. Now that we understand how growers choose their seeds, let's move on to understanding the differences between regular, feminized, and auto-flowering seeds, as this knowledge is crucial for making an informed choice that aligns with desired outcomes. Regular seeds are the classic choice for traditionalists and breeders alike. These seeds hold the promise of both male and female plants, typically in an even split. Their versatility makes them a favorite, allowing for natural pollination and the creation of new strains. However, growers must stay vigilant, identifying and removing male plants to prevent unwanted pollination that could lead to seeded buds and diminished quality at harvest time. On the other hand, feminized seeds are a game-changer for cultivators who enjoy simplicity and efficiency. These specially bred seeds guarantee female plants only, achieved through careful techniques that stress the plants into producing pollen. This pollen is then used to pollinate other females, ensuring the absence of male chromosomes, ideal for those aiming to maximize their yield of cannabinoid-rich flowers. Feminized seeds streamline the cultivation process, eliminating the need to cull male plants and allowing for more effective use of space and resources. Transitioning from the simplicity of feminized seeds to the unparalleled convenience and speed of auto-flowering seeds opens up even more possibilities for efficient cultivation. These seeds usher in a new era with plants that transition seamlessly from vegetative growth to flowering based on age, rather than relying on changes in light cycles. This remarkable trait stems from the inclusion of Cannabis ruderalis genetics. Auto-flowering seeds are a boon for growers seeking swift harvests, multiple yields per season, or those grappling with limited space and light control. Additionally, their compact size, abbreviated life cycle, and need for light make them a versatile choice for various environments. Once the ideal seeds have been selected, cultivators will then focus on the seed germination process. Let's discuss a few of the more popular methods, starting with the paper towel method. First, the cultivator will gather all the necessary materials: seeds, 2 paper towels, 2 plates, or a plastic container, and a spray bottle filled with water. With the materials in hand, the cultivator will start by moistening the paper towels, using the spray bottle to dampen the towels, ensuring they are moist but not dripping wet. Next, the paper towels are placed flat on a plate or inside the plastic container. The seeds are spread on the towel, making sure they're evenly spaced and not touching each other. Once the seeds are in place, they are covered with the second damp paper towel, ensuring they're completely enveloped. To create the ideal environment for germination, another plate is placed on top, or the container is sealed to trap moisture and create a dark, humid space that acts as an incubator for the seeds. The seeds should then be placed in a warm location with a constant temperature between 75 and 80 degrees Fahrenheit. Within 7 days, signs of life will start to appear. The seeds will crack open, revealing a small white root known as the taproot, which will start emerging. Another popular germination process is also known as direct sowing. This method simplifies the growing process by directly planting the seeds into the soil or growing medium, whereby eliminating the need for a separate germination phase. To help ensure success, growers will use high-quality seeds and routinely monitor the planting area for pests and diseases that can affect the seed's growing process. Don't forget that proper spacing between seeds is essential to prevent overcrowding and competition for resources. For cultivators who are interested in growing multiple strains, labeling or creating sowing zones can help keep track of the different seed varieties. The first step in direct planting is to select the right growing medium. Cultivators need a well-draining soil or a suitable hydroponic medium. Make sure the medium is rich in nutrients but not too compact, as this allows for good air circulation and root growth. Preparing the soil properly is crucial. To do this, the grower will loosen the top few inches and moisten it lightly to create a hospitable environment for the seeds. Next, the cultivator will plant the seeds into the growing medium. When using soil, the cultivator will create small holes about 1/4 to 1/2 inch deep. They will then place one seed in each hole and cover it lightly with soil, ensuring the seeds are not buried too deep. Once planted, water the seeds gently to avoid displacing them. It's important to keep the soil consistently moist but not waterlogged, as this will prevent the seed from germinating. After planting, maintaining the right temperature and light conditions is vital. Keep the environment warm, ideally between 70 to 85 degrees Fahrenheit. If growing indoors, use grow lights to mimic natural sunlight, ensuring the planted area receives adequate light for the seeds to germinate. Another effective technique for germination is the water soaking method. This process involves soaking the seeds in water for a period of time to soften their outer shell, making it easier for the root to break through and encourage germination. To begin the soaking process, the cultivator will prepare a bowl with enough water so that the seeds will be submerged. Next, the seeds will be placed into the water and left to soak for 12 to 24 hours, which will soften the seed coat and initiate germination. Make sure to check the seeds periodically, giving any floating seeds a gentle tap to see if they will sink. If the seeds continue to float, it can indicate they are not viable, but this is not always the case. Once soaked and soft, the seeds are then removed and planted in a growing medium, such as soil or rock wool, as soon as possible to support growth. Thank you for joining us on this comprehensive journey through the fascinating process of cannabis seed germination. We hope you found this video informative and helpful, whether you're a beginner or an experienced grower. If you enjoyed this video, please give it a thumbs up and share it with fellow growers who might benefit from this information. Don't forget to subscribe to our channel and hit the bell icon so you never miss an update on our latest cannabis cultivation tips and tricks. Feel free to leave any questions or comments down below. We love hearing from you and are always here to help. For more detailed guides and exclusive content, check out our website and follow us on social media. Thanks again for watching. Until next time, stay curious.